Dear viewers, this video is part 7 in the series on cancer, that is neoplasia, in the Medicine for Common Man series. Now, coming back to the discussion, anti-tumor effector mechanisms. The principal immune mechanism of tumor eradication is killing of tumor cells by CTLs, specific for tumor antigens. CTL responses against tumors are initiated by recognition of tumor antigens on host antigen presenting cells. Many cancers, particularly those that are rapidly growing, have a high fraction of cells that are undergoing cell death, either by apoptosis or necrosis. Dendritic cells and macrophages in the tumor microenvironment ingest tumor cells or released tumor antigens and migrate to draining lymph nodes. Here, they present the antigens to be recognized by naive CD8 positive CTLs. Dear listeners, by the way, this educational video is made possible by PrescribeOn, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribeon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Mechanisms of immune evasion by cancers. Immune responses often fail to check tumor growth because cancers evade immune recognition or resist immune effector mechanisms. Since cancer is all too common in persons who do not suffer from any overt immunodeficiency, it is evident that tumor cells must have ways to escape or evade the immune system in immunocompetent hosts. Several mechanisms appear to be operative. 1. Selective outgrowth of antigen-negative variants. During tumor progression, strongly immunogenic antigen-expressing subclones may be eliminated, and tumor cells that survive are those that have lost their antigens. 2. Loss or reduced expression of MHC molecules. Tumor cells may fail to express normal levels of HLA class I molecules, thereby losing the ability to display cytosolic antigens and escaping attack by cytotoxic T cells. It is a type of white blood cell. Note: human leukocyte antigen HLA. Human leukocyte antigen molecules are found on the surface of most cells in the body. Human leukocyte antigens play an important part in the body's immune response to foreign substances. They make up a person's tissue type, which varies from person to person. The HLA system is composed of three regions: the class I region for molecules HLA A, B, and C. The class II region encodes for HLA DR, DQ, and DP. The class III region for proteins of the complement system. 3. Engagement of pathways that inhibit T cell activation. Tumor cells actively inhibit tumor immunity by upregulating inhibitory checkpoints that suppress immune responses. 4. Secretion of immunosuppressive factors. Tumors may secrete several products that inhibit the host immune response. TGF beta is secreted in large quantities by many tumors and is a potent immunosuppressant. 5. Induction of regulatory T cells, TREGs. Tumors produce factors that favor the development of immunosuppressive TREGs, which could also contribute to immunoevasion. Thus, it seems that there is no dearth of mechanisms by which tumor cells can outwit the host immune system. Evasion of immune surveillance Overview. Tumor cells can be recognized by the immune system as non-self and destroyed. Anti-tumor activity is mediated by predominantly cell-mediated mechanisms. Tumors antigens are presented on the cell surface by MHC class I molecules and are recognized by CD8 positive cytotoxic T lymphocytes. The different classes of tumor antigens include products of mutated proto-oncogenes, tumor suppressor genes, overexpressed or aberrantly expressed proteins, and tumor antigens produced by oncogenic viruses. Immunosuppressed patients have an increased risk of development of cancer, particularly types caused by oncogenic DNA viruses. In immunocompetent patients, tumors may avoid the immune system by several mechanisms, including selective outgrowth of antigen negative variants, loss or reduced expression of histocompatibility antigens, and immunosuppression mediated by expression of certain factors like TGF beta by the tumor cells. Antibodies that overcome mechanisms of immune evasion involving immune checkpoints are approved for treatment of patients with advanced cancer and are most likely to be effective against cancers with a high mutational burden. Genomic instability. Genetic aberrations that increase mutation rates are very common in cancers and expedite the acquisition of driver mutations that are required for transformation and subsequent tumor progression. Although exposure to environmental agents that are mutagenic, example, chemicals, radiation, sunlight, is unavoidable, cancers are relatively rare outcomes of these encounters. This state of affairs results from the ability of normal cells to repair DNA damage, the death of cells with irreparable damage, and other mechanisms such as oncogene induced senescence and immune surveillance. Several mechanisms contribute to genomic instability in cancer cells. Noted already is how p53 protects the genome from potentially oncogenic damage by arresting cell division to provide time for repair of DNA damage and by initiating apoptosis in irreparably damaged cells. Cancers with loss of p53 function not only accumulate point mutations but also are strongly associated with aneuploidy, which may take the form of deletions, amplifications, and complex chromosomal rearrangements. These genomic aberrations may occur in cells with defective telomeres or may be created by other types of chromosomal catastrophes that lead to DNA breaks in multiple chromosomes. In the absence of p53 function, cells with severely damaged genomes that normally would be eliminated persist and stitch their chromosomes back together in an error-prone way using the non-homologous end joining pathway. Example, dicentric chromosomes discussed earlier. TP53 is the most commonly mutated gene in cancer, and loss of p53 function is thus the preeminent source of genomic instability in cancers. There are two other classes of proteins that normally function to protect against genomic instability. One, DNA repair factors, and two, DNA polymerase itself. As with p53, dysfunction of these factors leads to more rapid accumulation of genomic damage, speeding cancer development and progression. There is also a third type of protection against genomic instability specific to lymphoid cells that also is a source of oncogenic mutations. 3. DNA mismatch repair factors. DNA mismatch repair proteins work together to act as spell checkers during the process of DNA replication. For example, if there is an erroneous pairing of G with T rather than the normal A with T, the mismatch repair factors correct the defect. With proofreading function lost, errors accumulate throughout the genome. Some of these errors may by chance create driver mutations, and with time a cancer may result. One of the hallmarks of mismatch repair defects is microsatellite instability. Microsatellites are tandem repeats of 1 to 6 nucleotides found throughout the genome. In normal people, the length of these microsatellites remains constant. However, if mismatch repair is defective, these satellites are unstable and increase or decrease in length, creating mutated alleles. Note: allele is one of two or more versions of DNA sequence at a given genomic location. An individual inherits two alleles, one from each parent, for any given genomic location. If the two alleles are the same, the individual is homozygous for that allele. Trouble arises when cells acquire somatic loss of function mutations, presumably at random, in their single normal alleles. DNA polymerase. Under normal circumstances, cellular DNA polymerases involved in DNA replication have a very low rate of error, defined as additions of nucleotides that does not match its partner on the template strand of DNA. This fidelity stems in part from an inherent exonuclease activity that allows DNA polymerase to pause, excise mismatched bases, and insert the proper nucleotide before proceeding down the template strand. Certain cancers, most often endometrial carcinomas and colon cancers, harbor mutations in DNA polymerase that result in a loss of this proofreading function and the accumulation of numerous point substitutions. Cancers with DNA polymerase mutations are the most heavily mutated of all human cancers, and presumably because of a high burden of neoantigens appear to have excellent responses to immune checkpoint inhibitors. Genomic instability as enabler of malignancy. Overview. Persons with inherited mutations of genes involved in DNA repair systems are at greatly increased risk for the development of cancer. Patients with HNPCC syndrome, see below, have defects in the mismatch repair system leading to development of carcinomas of the colon. These patients' genomes show microsatellite instability characterized by changes in length of short repeats throughout the genome. Note: Lynch syndrome, also known as hereditary non-polyposis colorectal cancer (HNPCC), is the most common cause of hereditary colorectal, that is colon cancer. People with Lynch syndrome are more likely to get colorectal cancer and other cancers, and at a younger age, that is before 60, including uterine, that is endometrial cancer, stomach, liver, kidney, brain, and certain types of skin cancers. Patients with xeroderma pigmentosum have a defect in the nucleotide excision repair pathway and are at increased risk for the development of cancers of the skin exposed to UV light because of an inability to repair pyrimidine dimers. Note: Xeroderma pigmentosum, which is commonly known as XP, is an inherited condition characterized by an extreme sensitivity to ultraviolet rays, that is UV rays, from sunlight. This condition mostly affects the eyes and areas of skin exposed to the sun. Mutations in DNA polymerase that abolish proofreading function leads to genomic instability in colonic and endometrial carcinomas. TNB cells undergo regulated genomic instability during somatic gene rearrangements. Errors in this process are an important cause of lymphoid neoplasms. Cancer enabling inflammation. Infiltrating cancers provoke a chronic inflammatory reaction, leading some to liken them to wounds that do not heal. In patients with advanced cancers, this inflammatory reaction can be so extensive as to cause systemic signs and symptoms such as anemia, fatigue, and cachexia. Note: cachexia is a wasting syndrome that leads to loss of skeletal muscle and fat, is estimated to occur in about up to 80% of people with advanced cancer. Proposed cancer enabling effects of inflammatory cells and resident stromal cells include the following: release of factors that promote proliferation, infiltrating leukocytes and activated stromal cells secrete a variety of growth factors such as EGF as well as proteases that can liberate growth factors from the ECM. Note: epidermal growth factor EGF, located in chromosome 7p12, is a protein that stimulates cell growth and differentiation by binding to its receptor EGFR. Submaxillary gland and kidney are predominant sources of EGF production. EGFR signaling promotes embryonic development and stem cell regeneration. EGF plays pivotal role in ulcer-wound healing. Removal of growth suppressors. The growth of epithelial cells is normally suppressed by cell-to-cell and cell-to-extracellular matrix interactions. Proteases released by inflammatory cells can degrade the adhesion molecules that mediate these interactions, removing a barrier to growth. Enhanced resistance to cell death. Detachments of epithelial cells from basement membranes and from cell-to-cell interactions leads to a form of cell death called anoikis. Tumor-associated macrophages prevent anoikis by expressing adhesion molecules such as integrins that promote direct physical interactions with the tumor cells. Stromal cell cancer cell interactions increase the resistance of cancer cells to chemotherapy, presumably by activating signaling pathways that promote cell survival in the face of stresses such as DNA damage. Inducing angiogenesis. Inflammatory cells release numerous factors, including VEGF, which stimulate angiogenesis. Note: vascular endothelial growth factor VEGF is a potent angiogenic factor. It is an essential growth factor for vascular endothelial cells. VEGF is upregulated in many tumors and its contribution to tumor angiogenesis is well defined. Activating invasion and metastasis. Proteases released from macrophages foster tissue invasion by remodeling the extracellular matrix ECM, while factors such as TNF and EGF may directly stimulate tumor cell motility. Note: tumor necrosis factor TNF is a multifunctional cytokine that plays important roles in diverse cellular events such as cell survival, proliferation, differentiation, and death. As a pro-inflammatory cytokine, TNF is secreted by inflammatory cells which may be involved in inflammation-associated carcinogenesis. Evading immune destruction. A variety of soluble factors released by macrophages and other stromal cells may contribute to the immunosuppressive microenvironment of tumors. So, dear viewers, This video is part 7 in the series on cancer, that is Neoplasia in the Medicine for Common Man series. Thank you for watching the video.